as Dr. Sethu mentioned, I'm going to talk about circadian considerations during ischemic heart disease, and I do have some slides at the end about stem cells. So I just want to start off by reminding everybody that you are a markedly different individual at night versus the day. Okay, so clearly when you're asleep, you're very different from when you're awake. You have different levels of physical activity. You have different feeding status. You're fed versus you're fasted. And therefore you have marked rhythms in metabolism and various neurohumoral factors, as well as parameters such as cognitive function, and even at the cellular level, changes in signaling, in transcription. So at multiple levels, you are a markedly different individual. And that is absolutely true for the cardiovascular system. And this is a wonderful example of time of day dependent variations. So when you're asleep, your blood pressure, as long as you're a healthy individual, your blood pressure and your heart rate will drop. And then when you wake, you'll have this abrupt and very sudden increase in both of those parameters, and they will remain at an elevated level during the active period. And actually, you have a second peak. You can see here on this slide over on the right, you have a second peak also in the early hours of the evening as well. Now, associated with this increased heart rate and blood pressure in the early hours of the morning, you also have this increased incidence of sudden cardiac death and also the focus of the symposium, myocardial infarctions. You can see the zone of vulnerability right here is between about 6 a.m. in the morning and about 2 in the afternoon. Right here. So you have this marked increased risk of cardiovascular death and cardiovascular events. And then there is a second peak later on in the evening, which again is associated with this second peak in blood pressure. So if we were to summarize some of these findings that have been observed in humans, you can see that there are a large number of cardiovascular parameters that are peaking in the early hours of the morning. Blood pressure, heart rate, as well as adverse events. Not only myocardial infarctions, but also stroke, arrhythmias, and as I mentioned before, sudden cardiac death. And so you may be wondering, so what? Why should I care about this? Well, personally, I think that this is important because it's a golden opportunity for us to try and identify novel mechanisms for these various parameters. So, for example, for the initiation of a myocardial infarction. And so I don't know about you, but I like to think about scientists in terms of detectives. As you see here, they're trying to figure out what happened. And it doesn't matter if you think that you're a Columbo, or perhaps a Hercule Poirot, or Miss Marple, or Jessica Fletcher. She's always so happy. Maybe even Sherlock Holmes, or if you're a little bit of a character, you might be this guy right here. Not Gonzo, hopefully. I don't know about you, but I consider myself a little bit more of a Columbo. And so these individuals, what they're going to do is they're going to ask two questions when there's a murder. They're going to ask, first of all, what was the time of the murder? What was the time of death? And then, where was the suspect at that particular time? And what they're trying to do is they're trying to narrow down the different suspects, and then they'll do follow-up studies to try and figure out who actually caused the death. And so there are a large number of parameters outside of the cardiovascular system that are definitely contributing towards rhythms in both cardiovascular physiology and pathophysiology. So you have changes in autonomic, sympathetic, and adrenergic stimulation. You have changes in physical activity, vascular resistance, and as you saw a moment ago, blood pressure. Since we're not cows or we don't graze, instead we have meals, we have oscillations in feeding and therefore various neurohumoral factors associated with feeding fasting cycles, as well as various other bloodborne factors, including platelet activity. And so what a very clever chap, Mueller, did back in the 1980s was that he investigated all the possible suspects and looked to see the time of day variations in these suspects and how that correlated with the increased onset of myocardial infarctions in the early hours of the morning. And what he found was, as I mentioned before, is that blood pressure is elevated during the early hours of the morning, as is also platelet aggregability, okay, so platelet activation, as well as various other parameters as well. And so it led to this hypothetical model here on the right, in which he hypothesized that the increased shear stress associated with waking up in the morning and this sudden abrupt increase in blood pressure would then lead to a rupture of vulnerable plaques, which, if there's increased platelet aggregation at this particular time, will lead to a thrombus, an occlusion, and therefore myocardial infarction. Okay, so that seems like a plausible model. And so that was the model for quite a long time. So then where do we go from here? Well, there certainly are other players that are not considered here. For example, as we've heard in this symposium, there wasn't any immune cells on here, no MMPs or other factors as well. So there could be other factors that are at play. In addition, it doesn't quite tell you what are the mechanisms driving these oscillations in these various risk factors. In fact, we have to go all the way back to 1938 to some really seminal studies by this guy right here, Nathaniel Kleitman, who decided, wouldn't it be cool if we had a 28-hour day? Okay, wouldn't everybody love an extra 4 hours a day, all right, being able to cram in a little extra work? It would be kind of cool, especially near grant time. So what he decided to do was he decided to ask the question, can humans adjust to a 28-hour day? Is it even possible? Which, of course, might be important for future colonization of other planets that might have a 28-hour light-dark cycle. So what he did was he and his intrepid colleague right here, they went into these caves in Kentucky. These are the Mammoth Caves, back in 1938, and they took with them this clock that had a 28-hour rhythm to it. Okay, it's not a 24-hour clock, it was a 28-hour clock. And what they did was they forced themselves to live a 28-hour day by waking each other up. And so they went down in these caves with the clock, with some food, with some clothes, and they went down there for just over 30 days. It was 32 days. In addition, they took a nice little rectal probe as well to monitor their core body temperature. And what they found was, even though they were living a 28-hour day, which was extremely difficult, as they documented in their notes, they found that their blood pressure rhythms stayed at 24 hours. So it was completely independent to their sleep-wake cycles. It was something endogenous. And so when they came out of these caves, they actually considered their experiment was a failure. But it wasn't until about the 1960s when people realized that he actually uncovered the fact that there's an endogenous clock within humans that are driving physiology. And so it wasn't until the 1960s when Ashkoff performed similar studies in individuals that he kept under constant conditions, constant lighting conditions, and found that body temperature rhythms, as well as sleep-wake cycles and urine excretion, all had rhythms with a periodicity of between 24 and 25 hours. And so these really are being driven by endogenous circadian clocks. So what are these circadian clocks? Well, so here's the human circadian system, and when most people think about the circadian system, they think about some clock in the brain that is regulating sleep-wake cycles. And that's absolutely true. There is a clock within the SCN, the so-called master clock, which can control, can signal to the pineal gland and influence melatonin oscillations, which in turn can influence sleep-wake cycles. In addition, it's also through autonomic nervous stimulation can influence core body temperature, as well as various other neurohumoral factors. But it actually turns out that within every cell in your body, not just within the brain, but every cell in your body has an endogenous clock. And all the apparatus is found within each and individual cell. And there's a little bit of a hierarchy going on. That what happens is that when you wake up, light is sensed by a specialized ganglia within the retina, which will then reset your central clock, which then sends out neurohumoral signals to the clocks within peripheral tissues and resets those clocks. And it's these clocks then that are driving oscillations in various physiologic parameters such as body temperature. So the unfortunate part of this is that you can completely dissociate the central clock from these peripheral clocks by your behaviors, such as eating at the wrong time of the day. Because these peripheral clocks are very sensitive to neurohumoral factors, if you eat at the wrong time of the day, you can adjust the rhythms in neurohumoral factors, and in turn, you can dissociate your brain from your periphery. And that, 9 times out of 10, is associated with disease. So here is the molecular clock. I promised that I wouldn't show too many molecular slides, but this is one of only about 3 or 4. Just to show you that this is truly a transcriptional endogenous mechanism. It's found intrinsic within every cell within your body. And at the heart of the mechanism are these transcription factors, clock and BMAL1. And what they do is they can induce negative components of the clock that over a 24-hour period will increase feedback and inhibit the transcriptional activity of clock and BMAL1, so that you get these nice oscillations in clock BMAL1 activity. I only want you to remember clock, BMAL1, and period proteins, okay? Those are the only ones that I'm going to mention today. So with some very clever guys in Harvard, Steve Shea and also Frank Shearer did, and actually Steve Shea has now moved to Portland, Oregon. What they decided to do was they decided to do those experiments that Kleitman did. And they placed individuals in a 28-hour cycle. They forced them to live 28-hour days. And remember, if you do that, what you can do is you can uncover in humans endogenous 24-hour rhythms that are completely independent of your sleep-wake cycles, of your feeding-fasting cycles. These are truly driven by endogenous factors. And when they did that, they then looked at various cardiovascular parameters, including parameters that may increase your risk of having a myocardial infarction. And what they found was that various parameters associated with platelet activation, as well as thrombolysis, such as Pi1, these had endogenous 24-hour rhythms that are being driven by endogenous clocks. So they're not simply being driven by light dart cycles. In fact, they went on, and what they've done is they've looked at a whole host of possible candidate parameters that may be contributing towards increased risk of a myocardial infarction during the susceptibility window, as shown in the red rectangle. And what they found is certainly platelet activation and Pi1 are being driven by endogenous clocks. They are not being driven by your behaviors. And whereas things such as blood pressure, there actually is an endogenous rhythm, but it's peaking during the evening. It's that second peak. And that first peak in blood pressure is actually being driven by your sleep-wake cycles and not by an endogenous clock. So based on this, what we can, I wanted to show you that, again, this is my second slide of molecular data, just to show that, in fact, Pi1 is a directly regulated clock gene. So if you look in mouse tissues at different times of the day, here's heart and here's kidney, you can see that levels of this gene, the activity goes up, or the gene expression goes up, right at the time at which a rodent will wake up, because rodents are nocturnal. And so studies in human cells using the human promoter of Pi1, it was shown that clock and BMAL1 can directly bind to the promoter of Pi1, and this is responsible for the peak in Pi1 in the early hours of the morning. So when we go back to this model then of Mueller's, what we can say is that probably the increase in blood pressure and the shear stress that's inducing the rupture of these vulnerable plaques in the morning are probably secondary to environmental and behavioral changes associated with the abrupt increase in shear stress when you wake up and blood pressure goes up. However, the increase in platelet activation and the elevation in Pi1 seems to be driven by endogenous factors, by these circadian clocks that are found within your body. And therefore, it's a combination between the endogenous clocks and also your behaviors that are leading to the rise in myocardial infarctions in the early hours of the morning. So when these studies were published, I couldn't help but write a commentary on this, and I had to come up with a catchy title. Okay, and so I said that Pi1 at breakfast, whether you like it or not. Because you might think, well, it's actually, why did we evolve this way? Why would we want Pi1 to go up in the morning? It's not advantageous to have a heart thrombosis in the morning because you're going to die. Well, actually, if you think about it, it is advantageous to increase clotting in the morning. And so we have to remember, putting aside, of course, religious undertones, we have to think about what is the meaning of life. Well, the meaning of life is simply to survive long enough to pass on your genes. Okay, that's the purpose of life, to be honest, without the religion. And so, and when you wake up, what you have to do is you have to go out and you have to forage for food, you have to avoid predators, and you have to reproduce. And when you reproduce, you might have to fight suitors in order to gain the mate that you want to reproduce with. All of those things are risky to do. Okay? You might, if you're very hungry in the morning, you might take risks and you might fall. You might scratch yourself. If you're avoiding predators, you're running away from predators, if they catch you and they bite you, you're going to get hurt. And if you're fighting a suitor, you also could get hurt. So in the early hours of the morning, it's very important that you rev up the ability to heal. And so that's why there's this increased platelet activation in the early hours of the morning. At least that's the concept. And so these clocks, what they're doing is they provide this selective advantage of anticipation. They are predicting that you are more likely to injure yourself in the morning compared to other times of the day, certainly in comparison to when you're asleep. Okay, so that's one side of the coin, is the onset of a myocardial infarction. What about the tolerance to a myocardial infarction? Are there rhythms in that parameter? And indeed, these were some studies that we did back in 2010, and we published in Circ Research. We induced a heart attack in mice at different times of the day using the LAD ligation, and this is ischemia reperfusion. And what we did was we performed TTC staining to look at infarct size. And we found that depending on the time of day, there was about a 3 to 3.5-fold oscillation in the amount of damage to the myocardium. It was the same stress, just different times of the day. And when we genetically got rid of the clock within the heart, we completely lost these rhythms. So this is being driven by this endogenous clock mechanism. Since these studies, they have actually been replicated in some ways in humans. There have actually been 3 human studies to date that have all shown that in the early hours of the morning, there is an increase, or I should say a decrease in the tolerance of the heart to a myocardial infarction because there is greater release of factors such as CK and troponin I into the circulation. At least there are higher levels in individuals that had myocardial infarctions in the morning. And actually in humans, there seems to be, again, a biphasic pattern, similar to the biphasic pattern that we've seen in other parameters, including blood pressure. Okay, so where do we go from there? Okay, so I've said that the clocks within your body are certainly contributing both to MIs, to the myocardial infarction incidence, as well as to the tolerance to myocardial infarction. So it then leads to the question, well, if you were to disrupt your endogenous clocks somehow, which I told you is very easy to do, even by just eating. If you ate pizza at midnight, you would disrupt your clock in your heart because it's the wrong time of day to eat. So if we were to disrupt our rhythms, our endogenous clocks, is that, similar to Salvador Dali's picture here, is that going to lead to an explosion of the system and make things even worse? And we actually believe that this is the case. So when I think about disruption of clocks, the first thing I think of is shift work. Okay, shift workers definitely disrupt their circadian clocks, their circadian rhythms. And so back in 2010, a report from the Institute of Work and Health indicated that up to 1/4 of the U.S. population, the U.S. workforce performs some form of shift work. And that there is an increased incidence of injuries, of cancer, of preterm deliveries, gastrointestinal disorders, obesity, diabetes, and of course, cardiovascular disease, the reason why we're here. And mental disorders too, which might explain that look on that cat right there. So when you look at meta-analyses that have been performed in terms of myocardial infarction risk in shift workers, you can see, I think that this was just over 20 different studies, you find that there's about a 7% increased risk of myocardial infarctions in shift workers, night shift workers, compared to their day shift counterparts. So it's modest, but it certainly is statistically significant. But we can disrupt our circadian rhythms through a number of different ways, and there does seem to be an association with an increased incidence in myocardial infarctions. So in fact, if you think about it, every weekend you disrupt your circadian rhythms. Okay, so every weekend you get up a little later, I'm sure. You change your eating behavior. You probably eat a little later. You even probably go to bed a little later. And then on a Monday morning, when you figure out, oh, I got to go to work, you find that it's actually quite tricky to wake up on Monday morning. And people say they hate Monday mornings, and it's true. And so this study showed that those individuals who are working versus individuals who are not working, on a Monday morning, there's an increased incidence of myocardial infarctions compared to any other time of the week. If we move on and we look at monthly, you can see that there's this seasonal effect that actually, so during the summer months, the risk of having an acute myocardial infarction drops. And yesterday was the last day of September. This was the happy zone. And now we are starting to enter the not so happy time. And so there's now an increased risk of having a myocardial infarction during the winter months. Well, that could be multifactorial, all right? The stress of the holiday season, the colder temperatures. But certainly in the winter months, your circadian rhythms are not as strong because you aren't exposed to as much natural sunlight. And so that's actually extremely important for various animals because those diminished rhythms in your circadian clocks are very, well, not in yours, but in animals, are very important for reproduction cycles and are also very important for migration of animals, such as birds. They can sense that the timing of their clocks is diminished, and therefore that signals for those various parameters. And then seasonally, so this is one that I get phone calls quite often, and that's when we perform the spring forward and the fall back. So unfortunately, when we have this spring forward and we lose a 1 hour, what happens is that the Monday and the Tuesday following the spring forward, you get this increased incidence of myocardial infarctions, about a 10% increase in myocardial infarctions. And then when we fall back, you actually find that there's a decrease by about 10% in the incidence of myocardial infarctions. Well, again, this sudden, abrupt change in losing 1 hour, it takes a while for your endogenous clocks to catch up. So it leads to a circadian disruption. And in addition, also sleep disorders are going to lead to disruption of your circadian system and are associated with increased MI incidence. So here, insomnia can be associated with up to a 1.9-fold increase in myocardial incidence, depending on the severity, the number of symptoms of insomnia that you have. Individuals who sleep less, so this is just sleep duration, 8, of course, is optimal. And then as you sleep less and you go all the way down to just 5 hours a night, oops, not advancing, okay, just down to 5 hours a night, you again see about a 1.9 increase incidence of myocardial infarctions following that sleep deprivation. And then, of course, sleep apnea, again, disrupts your circadian clocks, and it's associated with an increased incidence of myocardial infarction. It can be treated with CPAP, and it's been shown now that CPAP treatment does improve the molecular clocks, even when you look at rhythms in immune cells. In fact, we do this to our patients. So this was a slide that was kindly donated by Tammy Martino, a close collaborator of mine, who likes to remind everybody that when patients come into the CCU, what do? We wake them up. We keep them under constant lighting conditions. It's noisy in there. They're constantly being given drugs. You disrupt their circadian rhythms, yet you're trying to get them to heal. And in fact, this was noticed by Eugene Braunwald, who is a famous cardiologist, that back in 1952, he said that when they admitted patients for acute myocardial infarction, whenever there was a bed available, they would actually try to put them as far away from the nurse's station as possible so they would avoid being woken up by the telephone. Okay, so he realized that patients did better when they weren't being constantly disrupted. And so what Tammy decided to do was she decided to perform this study in which she said, okay, well, let's mimic the CCU. Let's induce a myocardial infarction in rodents, disrupt their circadian rhythms by disrupting the lighting, and look at the outcome in terms of infarct size as well as contractility of those animals, of the hearts. And so I just want you to focus down in panel G. I'm sorry I have so much information here. But panel G shows that when you disrupt the circadian environment of those mice, infarct size was much larger compared to animals in which you did not disrupt the infarction. You didn't disrupt the environment. This was done after the myocardial infarction occurred. It was just done for, I think, 4 or 5 days after the myocardial infarction to try and mimic what happens to patients. And so one thing that Tammy's trying to push is that maybe CCUs of the future should be a little bit more aware of the circadian environment and try to maintain that environment and not to disrupt the patients quite as much. So this brings us nicely to chronotherapy. This is my last slide before I get on to the stem cells. And so this was an interesting study in which they looked at the timing of taking aspirin. Okay, so a lot of people are told, take a low dose of aspirin and it'll help reduce your risk of a myocardial infarction. Well, when people look at the kinetics of the aspirin in the circulation, in the blood, following ingestion, so this individual, for example, took the aspirin at 9 in the morning, and you can see this spike of aspirin in the circulation, which then decays, comes down, and then by the time the next morning they're in this zone of increased risk for a myocardial infarction, there is practically no aspirin left in their circulation to reduce platelet activation. However, in individuals who take aspirin just before going to bed, you see this spike in the aspirin while they're asleep. And so then when they enter this zone of vulnerability, they still have aspirin in their circulation. It can still reduce the risk of an MI. And so this led to a clinical trial. Next slide. Sorry, I don't know if it's died. Let me advance it this way. This led to a clinical trial in which patients, or actually these were healthy volunteers, were given aspirin either at bedtime or awakening, and they found that when they looked at COX-dependent platelet activation, they found that individuals who took the aspirin at bedtime, they had greater depression of platelet activation compared to the individuals that took the aspirin in the morning. And so I think that now these studies are going to lead to trials investigating whether this is going to reduce MIs or not, taking the aspirin in the evening. Okay, let's move on to the stem cells. So when I was asked to talk about stem cells, I was a little nervous because I am not a stem cell biologist, but it did force me to go into the literature, and I actually found a paper, a review article, that I co-authored on this subject. So I must know a little bit about it. And so I went through and I reread the article that was written a couple years back, and a number of the questions that were posed in this article was whether either time of day or the circadian clock is influencing processes such as stem cell differentiation and proliferation, stem cell survival, engraftment, and or even any kind of immune contribution. And really, we have to go back and we have to, I love this experiment here. We have to go back and we have to remember one of the reasons why clocks evolved. One of the reasons why clocks evolved was to gate cell cycle progression. The idea is that you want to keep DNA replication separate, temporally separate from DNA repair. The reason for that is because when life was evolving in the primordial soup, the ozone layer was very thin. And so when the sun was up, what would happen is the sun would damage, there would be a lot of UV radiation, it would damage the DNA. If you had DNA replication at the time at which you have DNA damage, it would lead to a lot of transmission of mutations to daughter cells. It would lead to termination of life. So instead what you want to do is when the sun is up, you want to rev up DNA repair and you want to keep DNA synthesis at a low. And then when the sun goes down, that's when you take the repaired DNA and you can replicate it. And so this is exactly what the clock does. And so a nice study was performed by this group a couple years ago, in which they took neonatal pups and they exposed them to UV radiation either during the daylight hours or during the night. And what they found was when they exposed them to UV radiation during the normal light cycle, there was absolutely no tumor growth at all. There was no tumorigenesis. And that makes sense because that's the time at which DNA repair is high, DNA replication is low. But if you take those same animals and you expose them to UV radiation, but now during the dark, what you found was there was this huge increase in risk of development of cancer in these animals. What does this have to do with stem cells and also the topic today? Well, if you look at cancer cells, and particularly breast cancer cells, you find that many of human breast cancers have mutations in the period proteins, in particular period 2. And so some studies were performed in which they took cancer cells that didn't have mutations in period 2, and they mutated period 2. So they genetically ablated it through siRNA, and they found that when you do that, particularly with period 2, that you get this increased rate of tumor growth. So if period 2 is gone, then suddenly the tumor cells can grow a lot faster. Well, is that true also for stem cells? And in fact, it's led to this concept that period 1 and period 2 are very central right here in the control of cell proliferation and also in differentiation. And so when you look at stem cells, what you find is that resident dormant stem cells, they do have clocks, but they're not very well coupled. They're not communicating very well with each other, and they're kind of at a random time. And it's believed that that's very important for them to proliferate and also to be able to differentiate into various cell types. And once they become differentiated, then the clocks suddenly are all synchronized and the clock is fully developed. In fact, if you look at the development of a normal rat heart, this was an experiment done back in the early 2000s, they found that at postnatal day 2, that many clock genes, including the period 1 and BMAL1, they were oscillating quite fine in the developing heart. However, as the heart, it wasn't until postnatal day 14 when PER2 was fully synchronized. So that's a time at which there's this rapid growth of the heart. And so the idea is that PER2 may be very, very, very important for both proliferation and also differentiation into cardiomyocytes, as well as even maybe other cell types. So if that's true, then you would think if you knocked out PER2, that maybe it would be beneficial in terms of myocardial infarctions. And that's exactly what was found. So this was a study back in 2010 by Virag et al., in which they took PER2 knockout animals and they performed a permanent LAD ligation. So they induced an ischemic event in them. And they found 4 days later that there was actually smaller infarct sizes, even though this was permanent LAD ligation. And there was greater vessel density in the knockout animals in response to this ischemic event. Now, they didn't go into detail on looking at the stem cells, but I'm sure that this is something that they're going to do. Okay, so just to summarize, hopefully I've convinced you that there are these marked time of day dependent variations in various cardiovascular parameters, including the incidence of myocardial infarctions. And this increased risk of MIs in the early hours of the morning are due to a combination of your behavior as well as intrinsic clocks, such as with platelet activation. In addition, these clocks are driving MI tolerance, time of day oscillations in MI tolerance. And if you disrupt circadian rhythms, that increases your risk of not only having a myocardial infarction, but also decreases tolerance to that infarction. And I do believe that focusing in on the circadian clock and stem cells may be important, particularly for PER2. And so with that, I'd like to thank you for staying, and I'll be happy to answer any questions. Thank you very much.
Go ahead. Okay, great talk, Martin. So yeah, I think you've clearly shown that that disruption of the clock can impose or can cause injury, increase susceptibility to injury. But has anyone looked at the other way? Like if you have some injurious event on the cardiomyocyte, that can then disrupt the clock and then causing a feed-forward kind of mechanism?
That's a great question. I didn't show those studies. It actually does happen. These are some studies that we published a while ago. We induced a myocardial infarction in rats. It was 30 minutes of ischemia followed by reperfusion. And we sacrificed the animals and took out the hearts every 3 hours immediately after that infarction. And we separated the border zone from the remote region and found that in the border zone, the clocks were completely disrupted within 3 hours. They just flatlined. It was, instead of oscillating like this, they just went down like this and they stayed flat. But in the remote region, you found that the clocks were still oscillating. So in the border zone, the clock is disrupted by a myocardial infarction. Yep.
Hello, Martin. It's very interesting. I'm totally convinced by your talk that we have Earth first, then so we have very strong biological clock, and we don't want to disturb it. Otherwise, we will get sick. My question is, do you have any opportunity to tell NASA investigators do not send men to Mars because they have totally different daylight cycle? If we do that, if they do that, and the astronaut will be killed by the early morning myocardial infarction.
Now, hopefully they won't perish that quickly, but. So yes, this is something that NASA is really interested in, is circadian biology and sleep biology, even for the astronauts that are in the space station. So it only takes 90 minutes to orbit Earth. So every 90 minutes, we have a light-dark cycle when you're in the space station.
But they follow the 24-hour cycle.
Yes, they do. So they use artificial lighting within the space station. And so colonizing different planets is something that they're very interested in. Now, if I remember correctly, Mars, its light-dark cycle is only about an hour or two different from Earth.
24 hours something, yeah.
Yes, exactly. It's only about an hour off. It's not that off.
But that's shift every day.
Right, yes. So it would be detrimental, especially because I actually think that would be more detrimental than when because what happens is if you go 4 hours outside of the 24-hour, then you're outside the range of entrainment. It means your endogenous clocks will not try to re-entrain. I actually think it's worse when every day your body is trying to re-entrain. And so if you go into an environment that, say, is only 22-hour light-dark cycle or a 26-hour light-dark cycle, that would be far more detrimental than having a 20 or 28 light-dark cycle. Because
our bodies follow the 24 hours. Actually, my question is, if they really contact you, do you have any advice for them? Because we can use that here.
Yeah, absolutely. I mean, they're actively researching this all the time. Folks are sending grants to NASA. I review for them, and I haven't submitted one myself, but maybe I should. And they're extremely interested in understanding, are there ways in which they can manipulate the clock? Because that's what people want to do now, is they want to be able to fix the clock, and they want to be able to entrain it. And so there have been some small molecules that have been identified in which they can reset the clock. The two targets right now that are hot are casein kinase 1 epsilon and reverb alpha. And when people do that and give animals, so far it's been animals, when they give animals these agonists and antagonists, because one is an antagonist, one's an agonist, you find that it decreases adiposity, it improves insulin sensitivity, it actually even improved atherosclerosis parameters. But nobody's looked yet in things like ischemia reperfusion tolerance, and that's something that we're hopefully going to be working on very soon. Yes.
Marty. So I'm first. Sorry.
I'm sorry.
It's a very nice review, and to answer Dr. Chen's question, maybe we can build a cath lab first, then send them. Okay. So just a joke. So when you mentioned, during the daytime, blood pressure increase. a shear stress increase. So you have this plaque rupture. So it causes this like blockage. So that explains why the incidence increase. But why the infarct size is bigger? Right? Does that have anything regulated with metabolism and also the susceptibility of the myocardium?
You bring up a great question. So, I mean, the easy answer is we don't know. We know that these circadian clocks are contributing towards infarct size, clearly. When we genetically ablate the clock in the cardiomyocytes, we lose that oscillation. So we certainly know that the cardiomyocyte clock is regulating parameters like metabolism, like autophagy that you're interested in, signaling as well. GSK3 beta, for example, was something we were very interested in for a while because that definitely is influencing ischemia reperfusion tolerance. So I think it's not going to be just one thing that the clock is doing, but it's going to be regulating a whole host of processes that are ultimately leading to that oscillation. And right now, we don't know is the answer. Great. Yes?
Yeah, I think we all have an experience with circadian rhythm with simply jet lag.
Yes, that's right.
And I wonder, has anybody looked at people who are employed in situations where they have to constantly go through time zones? Pilots, sea captains, people who travel. Is there any increased mortality from those changes in circadian rhythm?
Yeah, absolutely. So there is. So in pilots and also in the assistants, there is an increase in cardiovascular disease. I'm trying to remember whether the data out there is more for sudden cardiac death as opposed to myocardial infarctions. Off the top of my head, I can't remember. But certainly in pilots and also the attendants, there is an increase in cardiovascular disease. Thinking about time travel, not time travel, sorry, traveling through time zones. Time travel would be great. Thinking about traveling through time zones, there is one study that I always find fascinating, but I didn't mention it today because it really is focused more on sudden cardiac death. But I think it really shows the importance of these endogenous clocks. And it was a study that was performed on vacationers. And so it was actually performed in Hawaii. And what this study did was they looked at the time of day at which folks in Hawaii were dying. And what they found was that vacationers in Hawaii were not dying in the early hours of the morning in Hawaii. They were dying from sudden cardiac death at the early hours of the morning from their point of origin. And so it really shows that it's this endogenous clocks that are really driving not only cardiovascular physiology, but also these adverse cardiovascular events.
Just one observation as you were talking, I remembered an observational study from years ago about complication rates in angioplasty, and they tend to be higher in the morning than in the evening, when the angioplasty is done in the morning. Yeah. So if you're going to have one, might as well do it in the afternoon.
If you're going to have it, it'd be better. Thank you.
Just a quick question.
Sure.
Is there any recommendation time of exercise during this circadian rhythm? When would be optimal to exercise? Because if the early morning hours and blood pressure.
That's a good question. Now, I can answer that question in terms of healthy individuals. I would not want to have to answer that question in terms of prescribing exercise for someone who has cardiovascular disease. So it all depends on what you want. So if you want to build muscle, or you want to increase aerobic capacity. So you tend to find that your aerobic capacity is better in the morning and in the early hours of the day, whereas your anaerobic, your strength, is better in the evening. And so your response to weight training versus aerobic training is very different depending on the time of day at which you perform the exercise. And that's in healthy people. I don't want to say anything about disease patients.
Follow up on that jet lag question. You know, for people that travel multiple time zones, some take melatonin to. So does that have an influence on the circadian clock, melatonin? The other question was your seasonal variation. Did you see any spike with tax day and?
Yeah, there should be, right? Yeah, absolutely. For the tax day, I don't know. I have not seen any. There should be. Well, except some people might be getting a nice tax break, and so then it might even out. For your first question, I'm sorry, the melatonin, yes. So melatonin is very important for sleep, and actually we have Suzanne in the audience, and she knows better than I do about sleep. It's very important for helping with restoring sleep rhythms. The data on resetting your peripheral clocks, for example, and the clock in the heart, it's not so strong. Melatonin is probably not a synchronizer for a large number of the peripheral tissues. Instead, it's probably more centrally important. Now, it does other things. It's even an antioxidant. And so rhythms in melatonin in your circulation could be important even for myocardial infarctions because it's an antioxidant. But in resetting peripheral tissues, the evidence is very poor. Instead, there are hopefully going to be other compounds, like these small molecules that I mentioned. They can reset your peripheral clocks.
Great.